Okay, so this is a video that I am recording in order to, I guess, prepare you for what's to come in the summer. But more specifically, for me to have a go at a A-level paper and show you essentially the things that you should be aiming to do while you're writing the paper in the summer. So, how you're supposed to analyze questions, what you should be thinking when you're reading the questions, all of these different things. Excuse me. So I'm going to do this under timed conditions. That's the plan. So I'll start in just a moment, so I can vaguely keep track of how long I'm spending on each question. And I'm going to sort of talk out loud and complete paper. Hopefully what you realize when I do this, and I'll mark it afterwards, is that I don't expect to get every mark on the paper, and neither should you. You should obviously aim to do that, of course, but don't get hung up over question, 1 mark here, 1 mark there, when you're sort of completing sort of short or long answer questions. You only need 67% for an A star, or at least you did last year, so let's call it 70%. You can drop about 30% of the marks and still obtain the top grade. So don't worry too much about picking up every single mark. You should aim to pick up as many as you can, but there'll be occasional marking points that are just so strange that I call them write-offs. So I have chosen a paper 3 because this will assess everything from both year 1, so the lowest SIT content, and year 2. So and it's 1 hour 30 minutes. So let's crack on. Okay, cats are mammals. A light micrograph of a section through the trachea of a cat is shown in figure 1.1. Right, okay, so we're talking about trachea and gas exchange here. The fact that it's a cat, I don't think I really care too much. Without even looking at the questions, I immediately kind of looking as to what these labels might be and where I am in the cross-section of the trachea itself. So the thing that's pointing out to me where I am and kind of orientating me here is number or letter C, because these are quite clearly a sort of combination between the ciliated epithelial cells and the goblet cells. What that means is, if you know the structure of your trachea, sorry, my pen keeps highlighting the page for some reason, is that if this is your trachea, oops, I tried to get a shape there. There we go. You have the lumen of the trachea on the inside that is surrounded by the cells, ciliated epithelial cells, for example, here, that have the cilia on them like that, and occasionally you'll get a goblet cell, etc. Right, so this keeps going round, and the cilia point in towards the lumen. So the lumen is down here. Okay, so name part A. Part A is quite clearly a syncytium of muscle cells. You can see the sort of striated appearance. So you just need to know what type of muscle is in the trachea. So smooth muscle, okay, under the control of the autonomic nervous system. B, so this is a little bit more tricky. I think, certain weaker candidates would just assume that's the trachea because it looks like an open space. Hopefully something that puts you off saying that is the fact that, A, there's another one. So there's sort of 2 of these shown here, but also the fact that the lumen is clearly at the bottom. So this is actually within the muscular sort of layer. So this is cartilage. Okay. Right, name the 2 types of cell in the box labeled C in figure 1.1, outline their functions. Right, so we've got ciliated epithelium or epithelial cells. Technically, they're columnar. Okay, so as opposed to either cuboidal or squamous, but that's probably slightly out of the spec. The function, of course, is to beat the cilia in order to waft, which, believe it or not, is a term you can use, mucus towards the larynx or the mouth. The second one I've mentioned is the goblet cells, and these secrete mucus to trap particles. And I'm going to define what I mean by particles here, e.g. dust or microorganisms, so things like bacteria. Right. Alveoli increase the surface area to volume ratio. That's true because they're increasing surface area. So this is about Fick's law here. An alveolus in the lung of the cat has a spherical shape. Okay, so again, I'm going to sketch my circle. The diameter is 0.13 millimeters. Okay. This is what I would do in the exam, by the way. Oops, sorry, 13313 even. Read the question. The surface area is 0.053 millimeters cubed, and I need to calculate the surface area to volume ratio. Right, well, I've got the surface area here, so I need to calculate the volume. It gives me the equation. That's quite nice of them. I would ask that you learn that equation, but so volume is 4/3 pi r cubed. So I'm just going to plug my numbers in, and then I'm going to cube the radius, which is half of the diameter, of course. Okay, so I'm going to plug that into my calculator. So obviously this is 0.0, what would that be? 0.065 technically. So this comes to 0.00115. I'm going to put units there just in case. So that's the volume. And don't forget, the key thing with surface area to volume ratio is that the volume we always take as 1. Okay, so it will be something x to 1. So in order to make my volume 1, I need to divide it by itself, which means I then need to divide the surface area by the same factor. So I'm now going to do 0.053 divided by 0.00115, which gives me 46. So 46 to 1. Don't forget the to 1 bit. Explain why the large surface area to volume ratio of alveoli is an advantage to mammals. So the key thing is to explain, and it's within the context of mammals. So I am going to say that increasing the surface area to volume ratio increases the rate of diffusion. It's only 1 mark, so it's probably not going to want much about Fick's law, but I'm going to put here Fick's law. I have not said, just while we're going on this, I have not said that the alveoli have a large surface area. That is because it tells me that in the question, so I'm not going to get marks for puking that back up. So increases the rate of diffusion. I know it's only 1 mark, so that might even be the marking point. I don't know where the marks are going to come from, though. So I'm going to relate it to mammals, and I'm going to say that, therefore, I can have increased diffusion of oxygen into my tissues, or, well, into the bloodstream in the alveoli, across the alveoli, I should say. This is for increased, I shouldn't do that, increased respiration for increased ATP production. And this is important because mammals have a high metabolic rate or demand. Now, that may seem, and I admit, like quite a lot to write for 1 mark, but the reason I'm writing all that is because I don't know where that mark is going to come from. Bit of an odd question for just 1 mark, I think, considering you're having to explain. So it'll be interesting to see where the mark is for that. But the fact that I've written that much gives me confidence that at least, hopefully, I'll have picked up that mark. Right, onto the nervous system now. So we're still talking about a cat. So the nervous system coordinates a response to stop objects becoming stuck in their throat. They have sensory receptors at the back of the throat, that detect a stimulus of a large piece of food or a ball of hair pressing on the tissue, and the response is shown. Right. Few interesting things here before I even look at the question. So we've got our stimulus, that's fine. That is obviously detected by a receptor, which isn't mentioned in the diagram at least, which will be at the end of a sensory neuron, a sort of dendrite at the end of a it will be in or part of a dendrite of the sensory neuron. This then goes to the medulla oblongata, which is in the brain stem. Interestingly, we're not using the sort of full brain here. So brain question mark. This is just in the spinal cord. Well, it's right at the bottom of the brain, I guess. So it is part of the brain stem. Okay, we then go to relay neuron, fine, which goes to somewhere else okay, so it goes to nucleus ambiguous in the medulla oblongata, motor neuron, and then we get a response. Okay, so explain 2 conclusions that can be drawn. So I need to explain, and it needs to be in the figure of 1.2. So immediately I'm thinking, well, I've only got 3 neurons involved, which is typical of a reflex arc. So I would say this is a reflex action. Therefore, it is fast and involuntary. And the evidence I have for this is that only 3 neurons are involved. I'm going to put as a potential third, there's a sort of lack of higher brain function required. I'm going to question mark that one. I'm not really sure whether that's going to be on the mark scheme or not, but why not put it down? Vitamins are molecules that are consumed in the diet of animals and have essential roles in the body. So we can use TLC to separate a mixture of the vitamins, fine. Most of the vitamins are colorless. Suggest 1 method for visualizing the vitamins in TLC. So if they're colorless and you're using TLC, interesting. Interesting. Suggest 1 method for visualizing the vitamins in TLC. I would possibly go down the route of saying so everything fluoresces essentially under UV light. It's just to the degree and the exact, I guess, emission spectrum. So I'm gonna say shine under UV light, or shine. No, shine UV light and assay the fluorescence. However, to be honest, I'm not quite sure on that one, because I guess you could also dye you could like stain the vitamins somehow by adding a dye. That could also potentially, but I guess if it's a mixture of vitamins, maybe that's difficult. Although I guess you wouldn't have to stain them all separately, because they're going to migrate to different places on the in the stationary phase. Yeah, so maybe adding a dye would be better here. For the sake of 1 mark, I'm going to stick with my answer. I'm potentially a write-off here. But we shall see. First one to look at when I'm marking this, actually, that is slightly interesting. Right, we need to calculate the RF value of vitamin B2. Right, I'm on a surface here, so this will be a little bit difficult. I'm going to try and do it with a ruler. This might slow me down a little bit, but if I can, right, there we go. So I'm going to draw that. Can I draw a line on here now? Oh, no. Oh, that's annoying. So I can't draw a line. While the ruler's on there. Is that right? That's slightly annoying, isn't it? Okay. Why is that not working? Let's try it like this. Oh, I can. There we go. Perfect. Okay, so I'm going to take the midpoint of vitamin B2. That is that. So I now need to measure from the origin. So I'm going to line that up. This might be slightly out. Here we go. So that's what's that? 5, 10, 15 sort of arbitrary units from here to here. Obviously, in the exam, you'd use an actual ruler and do it in millimeters or something, but I've got no scale on this, so let's call it arbitrary units. So I need to come back again here. Right, so that was 15. So to the solvent front, it's 5, 10, 15, 20, 25, 30, 35, 40, 45. Ooh, is that 50? It's not quite, is it? It's so close on 50 that I think we'll call it 50. That might be because it's on the surface, I think. Okay, so I don't need my ruler now, so I can get rid of that. Right, so for the RF value, don't forget, it's the distance migrated by the particular spot that we're looking for, and it's the middle of the spot that we need to take the value from, and then that's divided by the solvent front distance. So I'm going to do 15 divided by 50. So I'm just going to double check, make sure I don't make any mistakes here. So 15 divided by 50 would be 0.3. Yeah, 0.3. So RF value 0.3. Vitamin B9 is needed for the synthesis of molecule D, which is shown in figure 2.2. And it looks to me like a bit like a nucleotide. In fact, very much like a nucleotide, slightly, yes. Although there's no phosphate group, but molecule D is a component in the structure of DNA. Oh, right, okay, so it is a nucleotide, fine. Okay. Interestingly, they're not telling you what the base is, and the fact that it's one ring, it's a 6-membered ring, has the more simple structure, so therefore it's got the more complicated name, so it's a pyrimidine. So that means it must be a cytosine or a thymine, and it's DNA, so it can't be a uracil. And you're probably not expected to know which one it is, particularly. I happen to know that is thymine. Yes, that's thymine, but you're probably not required to know that, just that it's a pyrimidine. Right, deficiency of this vitamin in the diet can cause interphase. So they're mentioning a key word here, to stop in some cells. So we're going to have to talk about the cell cycle. So just to draw that in here, again, I'll do this in the exam. I'm trying to do this under vague exam timings, although I am explaining things as well. Okay, so why would deficiency of essentially a nucleotide cause interphase to stop? The answer is, and again, if you've drawn it out, it should be obvious. If you start here and work your way through, when you get to S phase, that is where we do DNA replication, which is where we need nucleotides. And of course, if we don't have any nucleotides, because we have a deficiency of this vitamin, so we don't have much of this, that will cause our cell cycle to arrest in S phase and therefore not move into G2. So I'm basically just going to say that. I'm going to say molecule D is a nucleotide that contains thymine in DNA. If depleted, and this is where the link to interphase comes in, S phase cannot proceed, as DNA replication, or DNA cannot be replicated. I'm then going to say that the cell cycle stalls in S phase, so the S to G2 transition is blocked. There we go. Right, vitamin B2 is used by the body to make FAD. Right, okay, so the fact that there's a massive mitochondria in there also makes me wonder this is now this is a replication-based question. And it's also used to make, vitamin B2 and B3. Which one was this one? Oh, B9, right, okay, never mind. Okay, so we don't need to know the structures, that's fine. So 2 different vitamins now, just to be clear on that. Very, very good exam technique is to, not mention the vitamin above because it's a different thing. Okay, label figure 2.3 to show the locations where FAD gains hydrogen atoms and where FAD releases hydrogen atoms during respiration. Oops. Right. So essentially, this is asking where do your coenzymes work? And of course, we gain hydrogen atoms, both FAD and NAD will gain hydrogen atoms in the matrix in the Krebs cycle. Oops. Of the mitochondrion, and it will release them if you imagine the mitochondrial membrane, of course, with our protein carriers. Oops. Like that, and it will donate them to the carriers. So reduced FAD will then become oxidized FAD. This guy becomes reduced, and that happens in the inner mitochondrial membrane. So I'm going to be very specific here. Use the letter E to show where FAD gains hydrogen atoms. That is in the matrix. Be clear that's an E. And then F, I'm going to be very clear here. It's not in the intermembrane space. It is the protein carriers genuinely are located on the inner mitochondrial membrane. So I'm very clear here, and I'm going to put F right there. Right, we need to tick whether these are true or false by the look of it. Add a tick where it's correct and a cross where it's incorrect. Again, good exam technique. Actually do what it tells you to. Don't just leave the crosses blank and assume. Right, FAD and NAD is a prosthetic group. Ooh. Well, NAD isn't. They're both coenzymes. So they could be prosthetic groups, but they are transiently associated with the enzymes, the sort of dehydrogenase enzymes that pick up the hydrogen from molecules. Although I am thinking twice about FAD because it can. There are certain circumstances where it is classified as prosthetic as opposed to just being a sort of transiently associated coenzyme. I'm going to go with no because that's generally more true, I think. Certainly for OCR. Is reduced in the link reaction. Right, well, FAD isn't used in the link reaction, regardless of reduction or oxidation. But I'm going to pick up on the fact that we're talking about reduction here. So that means it has to pick up hydrogen. And absolutely, NAD does act in the link reaction to pick up some hydrogen when we decarboxylate pyruvate into acetyl-CoA when we add coenzyme A as well. Next one, oxidizes molecules in the electron transport chain. Again, oxidizes molecules in the electron transport chain. So just be clear on this. So I'm going to do a quick diagram. My electron transport chain, here we go. So my FAD and my NAD that are both reduced will give electrons up to my carriers and become oxidized. And then the carrier becomes reduced, not oxidized. So the electron transport chain molecules, i.e. the protein carriers, become reduced, not oxidized. So neither of those is true. Nice question, that one. Although apart from the FAD one, that'll be an interesting one, I think. ATP is produced in respiration. Some of these molecules are used to release energy for processes. For example, ATP is used in the early stages of glycolysis. That's true. So ATP is both required for respiration and produced in it. Thankfully, you'll never be asked which one came first. Suggest one other way in which ATP contributes to the process of respiration. Okay, so basically this question is saying where else do you need ATP aside from glycolysis? So I guess you could say in the Krebs cycle, although technically it's GTP, but never mind. I think probably the nicest one, just going in order, is the active transport of pyruvate into the mitochondria or the mitochondrial matrix, of course, for the link reaction and the Krebs cycle. Vitamin C affects the activity of enzymes in different ways. It acts as a coenzyme. Okay, lovely. For several enzymes in the synthesis of collagen. Okay, potentially this could be a lower six topic coming up for collagen structure, or not. Outline the role of coenzymes in biological reactions. Right, okay, this is basically saying what do coenzymes do. So I'm going to make it clear that I know that they transiently, they're not prosthetic groups in this case. Well, I guess technically they're a subset, but they associate. Actually, I'm not going to say transiently because coenzymes could include prosthetic groups. It's not telling me. So associate with, actually, it's saying what coenzymes are. So coenzymes are organic molecules. So just to distinguish them from the metal ions, cofactors, organic molecules that associate with enzymes and allow them to carry out their function. And we're not asked to give a specific example, but what I am going to say is how they do it, which is they one way in which they do it is they help to decrease the activation energy barrier of a reaction. That's probably enough. So the example that we came across where, in my lessons, of course, is magnesium ions as cofactors that help do this, but coenzymes do it as well. So that hopefully should be enough there. We're not asked to give a specific example there. A scientist investigates the effect of vitamin C on the activity of adenylyl cyclase. Okay, so we've come across this in upper sixth, especially for homeostasis, and it catalyzes the conversion of ATP to cyclic AMP, which of course acts as a secondary messenger. Here we measure the activity of adenylyl cyclase, so essentially the rate of reaction, without and with vitamin C, essentially 1 millimolar there. Okay. Oops. Right. So I'm going to be clear here which one's with vitamin C. So with vitamin C, I'm going to highlight that just to make it clearer for me to see. Right. Explain. So I need to explain what can be concluded from the results in figure 2.4. So I have to use the figure about the effect of vitamin C. Okay, interesting question. Okay. I really want to describe here what's happening, but the question is really asking me to explain about the conclusion as opposed to talking about why the curve looks like that. So hopefully what you appreciate is that this is it's slowing down the activity of adenylyl cyclase compared to normal. So it's acting as an inhibitor. And the type is tricky to work out. I would be tempted to say that this is a non-competitive. I'm going to put a question mark there. The reason being, it looks like this is going to tail off and keep going, sort of plateauing. But I would argue we don't have enough data, which is interesting because it's wanting us to sort of pass comment. I would want a bit more data here to confirm that this is going to plateau, because it could then start to increase, I guess. Unlikely by the way that this looks. Maybe that's enough, I don't know. But right, so I'm going to say it acts as an inhibitor. But I need to explain it. So the explanation is that the rate or the activity of adenylyl cyclase is lower at every ATP concentration. I'm going to question, is it a non-competitive? And my explanation here, of course, you need to know your enzyme graphs, is that Vmax is not reached. But I'm going to question mark this. Is there enough data? That hopefully should be enough for 2 marks, I think. What I'm not going to do is, of course, waste time describing why the curve looks like that, because that's not what the question is asking. Yeah, okay. That is one of interest, I think. I think I'm going to star that one. That is one certainly of interest to me. I think I've at least got 1 mark there. I think the second mark potentially a write-off. Depends on what they want exactly. Right, gosh, lots of words here. Genetic modification and selective breeding can be used to improve characteristics of crops. In high light intensity, photosystem 2 absorbs excess light. Okay, so this is about photosynthesis. So I'm just going to do a quick diagram here for my own benefit. So photosystem 2, and we've got light energy coming in. like that. But if I have excess light energy over here, what it's saying is it can take that and it can convert it into heat energy. And that is achieved by NPQ, and I don't care what that stands for. NPQ happens here. Okay, so just to be clear, I'm going to highlight that. And it converts excess light energy into heat. NPQ can continue when light intensity is no longer in excess. Okay, so actually, it not only can it take that, it can take some of these guys and convert them into heat as well, which is not a good thing. So that reduces the efficiency of light striking chlorophyll in photosystem 2. We can genetically modify crop plants to limit NPQ, fine. And we're showing a table here. Okay, so I'm just going to look at the table quickly. So we have 150 seconds after we decrease light. Okay, and we have NPQ, the unmodified plant, it goes down to 0.35. It gets lower, so that's a good thing. This is much lower. We have less NPQ. And of course, if I have less NPQ, that means more of these guys, more of these photons of light can strike photosystem 2. So that's good, because I can obviously produce ATP and reduced NADP, which can then lead to the Calvin cycle. So I might just put that actually, increase Calvin cycle. So that's a good thing. Rate of carbon dioxide fixation, on the other hand, the GM crop fixes more carbon dioxide. Right, okay, I mean, partly probably explained by that, I think. I think they sort of link together quite nicely. But of course, if we fix more carbon, we can have more glucose, more respiration, and more growth and all this sort of stuff. State 1 dependent variable, okay, rate of carbon dioxide fixation. Interesting, for 1 mark. So practically a freebie. Explain, okay, again, the effect of genetic modification on the rate of carbon dioxide fixation after 150 seconds. So I need to explain the effect on the rate of carbon dioxide fixation. So I need to explain this result, essentially. So why can I fix more carbon dioxide? And I think that links to what I said earlier about photosystem 2. And in the GM plant, the NPQ. So I need to talk about the NPQ. I think so. The GM crop has much lower NPQ value, and it's not asking me for data particularly, but I'm going to give it 0.25 arbitrary units versus 0.35 in the unmodified plant. This means that more. Hold on, let's read it again. It comes back to this diagram actually again. So it means if I have less NPQ going on, more of my photons of light can hit photosystem 2. More photons of light can hit chlorophyll, being specific, in photosystem 2. Actually, it's not just chlorophyll, it's and accessory pigments as well. In photosystem 2, this means that I produce more ATP and reduced NADP for the Calvin cycle, so I can fix more carbon dioxide, quite a lot more actually. Right. I think that's enough for that. I think that explains it quite nicely. I've given some data. I've talked about NPQ. Again, good exam technique is to realize that they're not going to give you something like this and talk about MPQ multiple times without them wanting you to make reference to it. So good nota bene there. Often a single company holds the patent for a GM crop plant, suggest a potential ethical issue that may exist if one company holds the patent for a GM crop. Don't know if I like this one. Ethical issue. If one company holds a patent for a crop like this. Yeah, interesting. So it's a suggest, meaning I am not expected to know. Trying to think. This is one of interest, I think, for me. Not sure explicitly where the mark will be or what they want. I guess it depends what they classify as an ethical dilemma. But it's got to be something to do with a company having a patent, so it's probably money related, but I'm not convinced example was like talking about money. I'm going to go for it, I think, and not worry about it too much. So potential write-off, it's 1 mark, not fussed. I'm not going to spend long on it. So I'm just going to go with it, I think. I can't think of anything better at this stage. So essentially, the company can charge lots of money for their crop, which, I don't know, it might make farmers poorer. It sort of exploits poorer, cash poor farmers. Yeah, potential write-off there. I don't know. It'll be interesting to come back to that. Right. Humans have been selectively breeding maize as a crop. Fine. Different varieties of maize have different genomes. Describe how bioinformatics and computational biology can be used to compare genomes. Okay. This is basically what is bioinformatics and computational biology. So I'm going to say bioinformatics can be used to store vast quantities of sequence or DNA sequence data for easy analysis by computers. Linking onto the computer thing, you can have coding people that can write computer programs or macros or do coding to compare sequence across thousands of genomes very quickly. What else can we say? This can be used to look for allelic similarities or differences. I guess this can infer relationships between them. Or look for important regions or genes. I think that's probably hopefully enough. Right, 2 characteristics that farmers have selectively bred in maize are color and fruit shape. Color is determined by a gene with, I hate it when they do that. So it says here, color is determined by a gene with 2 alleles, and that the allele is dominant. So big A is dominant to little a, and that's just not true. It really gets on my nerves when they do this. They do it all the time. It makes life easier for students, but alleles are just sequence of DNA. That's it. So you can't have an allele that's dominant over another allele. What happens is the big A allele encodes a phenotype that is inherited in a dominant way compared to the other allele. So really, what we should be saying is that purple is dominant over yellow as a phenotype. But anyway, that's just me being picky. Right, big A is dominant, results in purple color. So I'm going to write this down here just for my own benefit. So big A is purple, little a is yellow. We've got another gene. Ooh, okay. Big B is smooth shape, and little b is wrinkled. So we've got two genes here, and here we go. We are told that they are on different chromosomes. In other words, they are not linked. So there's no linkage. So the recombination frequency will be 50%. Okay, there is completely independent assortment. So that's fine. So there's nothing to worry about there. Right, so we're going to cross two plants. We've got a parent plant that's purple and smooth, and it's heterozygous for both. Okay, so it's going to be big A. So this parent is going to be big A, little a for the purple, for the color gene, and for the shape, or whatever you call it, the fruit shape, it's heterozygous again, and it is smooth. So it's big B, little b. Oops. Okay, so that's the genotype of that parent. The other one is yellow and smooth and heterozygous for BB. Right, okay. So if it's yellow, it must be little a, and it's big B, little b, because that's what we're told. So parental genotypes, big A, big B, little b, and little a, big B, little b. The gametes that are produced from these, so the way that you do this is you take the first one and you go there, so we can be big A, big B. You can then again go to the next one, so big A, little b. We then start at the second A, so we can be little a, big B. Or finally, we can be little a, little b. And they're gametes, so I'm going to circle them to show that I know that they are gametes, despite it also saying it there. So do that with the next one. So big A, little a, big B. We can be little a, little b, little a, big B, which we've got here, and little a, little b again. So actually, we're only producing two types of gamete for that. Right, we now need to draw a genetic cross. So I'm going to take this parent first. So I'm going to draw the gametes. So big A, big B, big A, little b, little a, big B, little a, little b. I'm going to spread that out a little bit. Oops. And I'm going to take the second parent down the side. So big A, little b, little a, big B, little a, little b. Okay, again, they are gametes, so I'm going to circle them. There we go. Right, so now we just draw them in. So here, I will do it in a different color just to make life easier. So I'm going to start with this parent on the top. So I'm going to go big A, then it's little a, and then it's big B, big B. Big A, little b, big B. Actually, yeah, let's keep it like that. Keep it simple. Little a, big B, big B. Little a, little b, big B. Down here, big A, big B, big A, little b, little b, little a, big B, little b, and all the littles, little a, big B, little b. Gosh, right, this is a lot of work for 4 marks. Expected offspring phenotypes. Oh God, we've got to. Right, okay. So big A, little a first. So if there's a big A, we're purple. So I'm going to highlight. Again, it helps to have colours, right? So we're purple here, purple here, purple here, purple here. Oops. Something has just happened to my OneNote. Don't know what's happened. I hope that we're now back. Right, so I've got to highlight this again. So essentially we are purple here, purple here, purple here, purple here, and all the rest are yellow. Again, have highlighters in the exam. So that's the color. In terms of the shape, big B is smooth, little b is wrinkled. So I'm just going to make that note. So big B is, I've forgotten already, smooth, right. So if I see a big B, we're smooth. So smooth, 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 wrinkled, smooth, wrinkled. Okay, so the expected offspring phenotypes. So we're going to have to write here. So you can be purple and smooth. We can be purple and wrinkled. We can be yellow and smooth. And we can be yellow and wrinkled. Now, fairly obvious. I didn't have to highlight all that. Hopefully it's fairly obvious that that would be the case because they are not linked. No linkage. Okay, we have completely independent assortment, so therefore you should be able to get every combination possible. However, it's nice just to prove it by highlighting them. The expected phenotype ratio, this is where the highlighting helps us. So I do it in dark blue. Right, so let's just count them up. So purple and smooth. I've got 1, 2, 3. Purple and wrinkled, 1. Smooth and yellow, that's another 3. And yellow and wrinkled, 1. So what I'm going to do is go 3 to 3 to 1 to 1, and I'm going to be very clear and say that this is purple and smooth. This is purple, wrong. This is yellow and smooth. This is purple and wrinkled. And this is yellow and wrinkled. 3 to 3 to 1 to 1 phenotypically. Cool. Quite a lot of effort there for 4 marks. Probably took me a bit longer than 4 minutes, potentially. Anyway. Oh, right. Conservation agreements. Lovely. Gosh, right. Okay, this came into effect in 1975. Here we've got the percentage of species threatened with extinction that have been protected. Right, okay. So I'm going to highlight the non-protected. I think that's the easiest, right? The rest are protected. And we've got trade. That seems to be the most efficient. way of doing it, over 50%. Although interestingly, I should have said actually 25%, just over 25% not protected, and they're threatened with extinction. Okay, no commercial trade doesn't work as well, it seems. Okay, the only thing I'd say is there's no stats there. Okay, so figure 4.2 shows the number of species assessed and the number of species categorized as threatened each year. Okay, so it's going to want us to dig this apart a little bit. First things first, I think it's hopefully obvious the gap's getting bigger here compared to like here and here. So as the years go on, I would say we're assessing more species than actually are threatened, or than are becoming threatened. So that's a good thing. What else can we say here? Total number of threatened species is still increasing. Still increasing, that's not a good thing. Fine. Is there anything else interesting about this graph? Not particularly, I don't think. Right. Oh God, 6 marker. Ooh, right. Okay. I'll just pick up as many marks as I can here. Okay. The aim is to monitor and regulate the international trade of threatened species to prevent extinctions. Yeah. So the only thing is that you should know, of course, is that this is not good enough by itself. Right. There are other ways to prevent extinction. You've got captive breeding programs, all this, all this other stuff. The law is one thing, but it's probably not enough. Well, as shown here, I guess. Right, use the data in both to evaluate how successful it has in achieving its aim. Okay, so I'm going to start with the pros. I think they're probably easier. So this is how I'd set it out. Pros. And I think I'm going to kick off with saying that that the trade requires export permits. I think I'll start with this obvious point. I think more species are being assessed than are becoming threatened. More species are assessed each year than are becoming threatened. The gap between assessed and threatened species is becoming larger. What else? Let's go back to here. What else is good? No misread that. Whoops. I was wondering. I knew something was off. Right, no commercial trade allowed. So that is a good thing, I guess. In that, we've got what here? 50, what about 20? What's that? Maybe it was 15. Maybe that's about 20 to about. What would that be? Threatened species. 20. So we're not allowing commercial trade for what, about 20%? I don't like the fact there's no, about 20 maybe? That's about 15 there. Actually, no, about, yeah, maybe. Oh, that's annoying. I don't know how to give that as a number. Oh, well, I'm not going to spend a huge amount of time on it, I don't think. So I'm going to say about 20-ish. I don't like the fact that I can't give a good number for that. It's under 25, isn't it? Yeah, about 20%-ish of the species that are threatened can't be traded. Okay, according to that. I think that's quite good, or commercially traded. Okay, I'm going to move on from that thing. What is it, 6 marks? Yeah, okay. So I've said three pros there. I'm going to move on to cons. And the obvious one is that there was quite a few of them that weren't protected. So just over 25% are not protected. Just over 25% of species that are threatened are not protected. That's bad. So I guess you can still trade 50%, right? Oh, well, actually, I guess you can trade all these, can't you? So what's that? 20 plus 50, 70% you can trade. So you can trade all of those and those just with either permits or not. So actually, the bad thing is you can still trade about 70% of threatened species. Yeah, not enough as well. Yeah, that's it. Forgot to put that. Laws, so conservation agreements. Alone are not enough to stop species becoming extinct. I think I've probably said it, well, hopefully said enough for 6 marks there. I'm now going to move on because I want to make sure that I have enough time to finish the paper. I can always come back to this if needed, but I've got enough there. And I'd spend another 5-10 minutes, picking stuff out of these graphs. So I'm going to move on, I think, and hope for the best. Right. Right. The kakapo, however you say that, never heard of that. It's an endangered species of bird. Population size experiences a large decrease, blah, blah, blah. Stated term for a large decrease in population size that reduces the gene pool. Okay, so this is absolutely a genetic bottleneck. Adaptations can be categorized into 3 different types. Right, list the traits, type of adaptation. Right, active at night, that's not anatomical, it's behavioral, it's not physiological, is it? No. So, behavioral. Green feathers for camouflage, that is anatomical. Slow digestion, physiological. Strong beak and claws, again, that's anatomical. Good, quite easy. So again, I spent a bit longer on that genetics cross and these conservation agreement questions, but I've picked up there. There's 3 marks there, so I could, this is technically worth 3 minutes of exam time. So I've sort of saved myself at least a couple of minutes there, I think. Oh, right. Okay, lots of reading here. Not all of it will be useful, though, I suspect. Right. They evolved from a common ancestor. There we go. So there was some speciation event here. So some... Oh, okay. Emergence of Alps. Right, there we go. So that's like a geographical barrier. Okay. So yeah, fine. And there's been another speciation event here within the mountain range, though. So that might be a different speciation event. Okay. So I think so this is sort of allopatric speciation here, and I suspect this is sympatric. Question mark. Right, the kakapo can't fly, it forages on the floor. Okay. The kaka can fly, and the kia can fly as well. Oh, interesting. Okay, so maybe this event is where they flew to different parts. Perhaps both of these can fly. The kakapo can't, so that kind of makes sense. Again, with this being a physical barrier in the first instance. Lives in mountainous forest habitats. All three species live on the South Island and had overlapping ranges until the population size decreased. Populations live on the north. Populations of kakas also live on the north. So here they maybe migrated north. Okay. A student studied the information and suggested that all three species evolved by sympatric. No. So disagree. Because evidence against sympatric is that it's allopatric instead, due to the emergence of mountains. What did they call it? Southern Alps mountain range. So due to the emergence of a mountain range, and we said that the kaka can fly as well. Can the kaka fly? Yes. Okay. So. Are they on the South Island? Yes, they are. All three species live on the south. Right. Okay. How do I explain that? Okay. So mountain range. This means that they had a geographical barrier. And occupied different niches. However, they flew back to the South Island because that explains why they're still there. Okay, but this is the thing with this type of question. It's evaluate. So I need to give some agreement here. So what evidence do we have? Well, we do have evidence for sympatric. What would I say here? Read the question again. Leaves, fruit on the ground, can't fly. Yeah, okay. So the evidence for sympatric is that they occupy the similar niches. Favorite niches, forest floor, for example. Therefore, they were isolated by I guess like behavioral niches. Yeah. Let's go for those 4 marks. Yeah, there's a lot of words here, making it a bit weird to. Right, let's go with that. Yeah, I'm going to say if there's a 4th mark there, write off just for now because I don't know what the time is, but need to move on. How many more questions have we got? Couple. Right. New Zealand has a high species diversity. Right, explain. Okay, right, easy one. 2 marks. I can save a bit of time here. So species richness is the number of different species in a community or ecosystem. Okay, just a definition that you need to pick back up. Whereas the evenness is the relative numbers, or the abundance, I guess, of individuals within a species. Well, no, not within a species. Of each species. Oh no. Yeah. How do we? Yeah. Even this is relative numbers of individuals. Well, yeah. I don't know how to word this. Within each species group, let's put that. In a given community or ecosystem. Okay, so as opposed to the number of different species, it's the relative number of individuals in each species, basically. Right, saved a bit of time on there, hopefully. Have a quick look at how many to go. Ooh, oh, there's a few left. Oh, no, end of question paper. Right, we've not got that long to go, which is good. Right, yeast can respire aerobically or anaerobically. Right, okay, seems quite straightforward. So I've got time and volume of CO2 produced. Right, I've got 2. Right, okay, I'm going to actually skip over this and see what I've been asked to do, and then highlight the relevant bit. Compare glucose and maltose. Right. So glucose in blue. Again, this is where highlighters work quite well. So it's the square. Is that square? Yes. As opposed to, was it maltose? Yeah, maltose, which is this one. Okay, nice and easy comparison. Immediately, I can see the maltose is consistently higher than the glucose. So compare the carbon dioxide production. So maltose CO2 production is consistently higher than glucose. My handwriting is getting worse because we're near the end. Anything else? And it gets bigger. The distance gets bigger. That's quite obvious. So over time, greater volume of carbon dioxide produced than glucose. Yeah, it says compare though, but they started at the same level, didn't? Oh no, we are consistently higher. Yeah, okay. I think that'll do. Right, the carbon dioxide production with fructose is approximately the same as with sucrose. Okay. Explain whether you agree or disagree. So interesting this. I agree here. Obviously don't agree there, and there's one point there where I agree, but aside from that, I don't really. But overall, I guess they end up being not too dissimilar. Yeah, okay. So what time point is that? I'm going to quote the data. 75, 77. It's halfway. What's that? 90? 2, 4, 6, 8. Yeah, it's about 90. Right. Okay. So 90, it's actually both about, what's that value here? So 1, 2, 3, so 2, 4, 6, 8 centimeters cubed at 90. So I'm going to say I agree, as it's 8 centimeters cubed at 90 minutes. But, and it's similar until about 20 or so, maybe. Until 20 minutes. But aside from that, which one's higher? Well, at the end, it's the triangle, isn't it? Fructose beats sucrose. But at the end, fructose has a higher CO2 production than sucrose. Again, I'm going to quote the data. So just shy of 20, 10, 12, 18 versus about 8 again. So 18 centimeters cubed versus 8 centimeters cubed. Right. Okay, so we're comparing the rate of anaerobic respiration of 2 species of yeast. Glucose is the respiratory substrate, same concentration of that. Make tenfold dilutions. Okay, suggest 2 ways in which a student can reduce the percentage error in their measurements when producing the glucose solutions. Okay, so this is when we're making the dilutions. So it's using a measuring cylinder to measure the volume of water and a dropping pipette to transfer the glucose solutions. Potentially use a volumetric flask to measure volumes. What else could you do? Tenfold dilutions from a 1 molar stock solution. Measuring cylinders measure the volume of water. Dropping pipette. Don't like the fact that there's a dropping pipette there. So I think use a graduated pipette versus the dropping pipette. There we go. Right, complete sentences. Okay, this should be right. 3 marks. We've got 3 minutes technically. To avoid contamination, place the yeast in glucose solutions produced using distilled water. Fairly obvious. Culture species of yeast in different flasks. Ensure anaerobic conditions by using like a flask with like a bung in it, right? So like a airtight or like sealed flask. Use the rate of carbon dioxide production to measure respiration rate. Standardize all other variables. Blah, blah, blah. Repeat the measurements with 10 populations. We can calculate 2 means and 2... Okay, so you've got the mean, so you'd calculate the standard deviation, probably. Of the mean, and then use a t-test to compare the two results. Nice and straightforward. Homeobox genes, Hox genes code for transcription factors that regulate the expression of structural genes. Fine. So structural genes as opposed to regulatory genes. So these are like enzymes and things. Describe the general roles of homeobox genes and suggest the roles in development. Right. Development of the brain. Right. So we haven't actually covered that specifically. So it's going to ask us to use our brains. General roles, of course, is that they are transcription factors that turn on expression of genes involved in regulating the body plan of organisms. Different Hox genes are turned on in different tissues. But the key feature of all Hox genes, in fact, is they are expressed in the same order as they appear in the chromosome, which is a key thing about Hox genes. They regulate two factors, don't forget. They regulate mitosis and apoptosis as well. So in the brain, why would this be important? I guess it determines like each part of the brain, each brain region, by causing transcription to be on and off in certain cells or neurons. What else can I say? I guess it puts the brain in the right place, right? Ensures brain and spinal cord are in the correct place, i.e. in the head portion of the body. That should be fine, probably. Right. Final question. I think we're just about on track for time. So scientists have compared structures of molecules between archaea, bacteria, eukaryotes. These are the three domains of life. Explain the conclusions drawn from the table about which domains are most closely related. Ooh, interesting. First thing that's screaming out at me is that the helicase is absolutely useless for this because they all have them. So useless. So I would say the helicase is useless as all domains have the same type. Otherwise, let's go for DNA polymerase. So SFB, SF. So archaea and eukaryotes share that. So archaea and eukaryotes are more closely related, as share SFB polymerase, whereas of course bacteria don't. Histones, of course, they also share. Bacteria don't have them. And the histones are also shared and absent in bacteria. Mitochondria doesn't really help too much, does it? I don't think. Ribosomes. Doesn't really help us, does it? Well, I guess we could sequence ribosomal RNA to infer relationships. And actually, it's the ribosomes that make archaea and bacteria look more closely related, even though they're not. Oh, right, we're near the end. Gosh, my handwriting is getting awful because my hand is hurting now. State the roles of DNA polymerase and helicase in cells. Right, hopefully fairly easy. DNA polymerase is to catalyze formation of phosphodiester bonds between adjacent nucleotides. And of course, helicases break hydrogen bonds to allow for DNA replication. Good, right, done. Whew. Right, I'm just going to flick to see what time we're on. We are on 1 hour 26 minutes, according to my clock. So if I scroll all the way up here, gosh, that's tough. So 1 hour 26 minutes, and I did talk a lot probably as well, rather than just getting on with it. So 1 hour 26 minutes. Gosh, 4 minutes to spare. Which just shows you, even for a teacher, and tough, right? I'm not convinced I've probably got every mark. So we'll see. Right. Okay, I'm going to pause the video. Something's gone really weird. Hold on. I'm going to pause the video and then I'm going to get the mark scheme up and mark it. Right. So I have managed to get the mark scheme up. So we're just going to mark this quickly and see how I did and where I lost marks. I'm assuming I did somewhere. Right. So smooth muscle and cartilage. Yes and yes. Good. Happy with that. Goblet cells secrete mucus. Yeah. And the ciliated epithelial cells move mucus. Yes. Good. So that is 2 there as well. Right. Fick's law. In fact, I'm just going to make that a little bit thinner. There we go. Right. So B 0.065 somewhere. Yeah. 0.01. So getting that as 2 marks, and then 46 is the third. So yeah, good. Happy with that. Oops, can't move that down for some reason. Right, more gas exchange or diffusion. Interesting. Okay, increased rate of diffusion. So I get the mark in the first sentence. All the rest is kind of irrelevant. Fine. Okay, no problems. Explain 2 conclusions that can be drawn: involuntary and does not use the somatic nervous system or coordinated by the medulla, or does not use decision making. So I have to say involuntary and lack of higher brain function. Would I get it for lack of higher brain function? Does not require conscious thought, happens without thinking about it. Huh. Allow the passes through the brain stem or the medulla. Okay, it wanted me to mention the medulla. I'm not going to give myself that. I get that one, but I don't get that one. So 1 there. So note to self: write the medulla. I know I talked about it. Write that in my answer. I'm going to be harsh there. Right, first one of interest. 2A. Stain or dye or exposed to UV light. Oh, lovely. And it does accept staining. Okay, that was a write-off that didn't need to happen. 0.3. So you get a mark for 2.4 divided by 8. I did it obviously in arbitrary units instead. Allow the answer for 2 marks. So there we go. Happy with that. Moving down. Oops. Right. D is a nucleotide or thymine. So I've talked about that, and a pentosugar deoxyribose. Well, I've mentioned thymine, so that got me the mark there. Fewer nucleotides or pyrimidine bases available. Yeah, depleted. S phase stops or is incomplete. Yes. Does not pass G2 checkpoint. I mentioned something. Yes. Yes. There we go. Easy. Nice. E anywhere in the matrix, so correct. F on the inner mitochondrial membrane, correct. Good. Hold on, how many marks have I lost so far? Just 1, right? So I'll keep track of that. Okay, so tick. Ooh, tick. Huh. FAD is a prosthetic group, but it isn't always. Oh, okay. So it says allow cross for FAD as a prosthetic. Right. Okay. So it accepts both. Okay, that's fine. So that's correct. Then it's cross tick and then cross. Right. Okay. Happy with that. Interesting because that's not on the spec and not quite true anyway. So, but I guess that's good on OCR for allowing both. Right. What have we got here? Active transport or synthesis of enzyme. Yeah, active transport. Fine. Right. Vitamin C. What do coenzymes do? They assist enzymes to carry out their function. Right. Okay. That's assisting enzymes without permanently being bound. Ooh, interesting. Decrease. Okay, example of mechanism. Right, that will do. For example of mechanism. Interestingly, they want you to be very specific and say they transiently associate. Thing is, prosthetic groups are a subset of coenzyme, really. Anyway. Right. Fine with that. So I've still only lost 1 mark, which is good. Axis inhibitor. Yes, 1 mark. Okay, non-competitive because increasing does not allow the enzyme activity to reach the level. Yep, good. I question that, interestingly. So it does mention non-competitive. It says allow non-competitive because Vmax isn't reached, so that's fine. So I got my 2 marks. Idea of cannot decide whether it's competitive or non because the enzyme activity is not plateaued. Okay, so potentially I would have also got a mark for saying about lack of data available. Nice. Right, we get rate of carbon dioxide fixation, fine. Right, greater rate because less light is converted to heat or lower NPQ. Yes, lower NPQ, that's 1 mark. So more electrons enter the electron transport chain. Didn't say that specifically. I just said that photosystem 2 can do its thing. So probably not enough detail for that. More ATP or reduced NADP generated for the Calvin cycle. Correct. Good. Note to self, explain the electron transport chain a bit more, perhaps. Yeah. Right. Ethical issue. Oh, yeah, this was an interesting one. Right. Yeah. High prices. Okay. So it was that. Good. Whew. Maybe not a write-off in the end. Not being available to everyone. Poorer farmers can't afford it. Right. Yeah. So it was money based. Interesting. I wouldn't have lost sleep over if I didn't get that, to be honest. Right. Sequence genomes. So did I mention DNA sequencing? Yeah, well, compare DNA sequence, yeah. Use algorithms or mathematical model. Well, I put macros and coding, but that's fine. Analyze differences or similarities in sequence. Yeah, okay, that's 3. Nice. Using software as well also counts. Well, that's the macro mark, actually, I guess. Yeah. Okay, nice. Right, parent genotypes, big A, little a, big B, little b. That's correct. And little a, big. Right, okay, so that's correct. 1 mark for those 2. 1 mark for the gametes, which are correct. 3 to 1 to 3 to 1 as a ratio. Okay, so the phenotype's purple and right. Okay, so that's correct and that's correct. Good. Still only on 1 mark dropped, which is good for question 4 even. Right. I didn't like this one. Didn't like this one. We might have messed up here. Right. Positive. The majority of species are protected. Majority of species identified as threatened are protected. Didn't say that specifically. Approximately 20% of the threatened species cannot be traded. Oh, yes, excellent. That was a last-minute addition. Number of species being assessed by the IUCN is increasing, which means more species can be added. Okay. I kind of said that up here, but I didn't explicitly say that means they can be added to the number of species being assessed is increasing, which means, yeah, I'm going to be harsh on that one. I think I'm going to say I didn't put enough detail in there. The number of species being assessed is rising faster. Right, okay, that's fine. The gap is bigger. Right, 2 marks for that one. Negatives, a large number aren't protected. Large number can still be traded. Yes, 70%. Trade is only one aspect of protect. Yeah, good. And that's probably it for me, I think. So I got 5. So I've lost a second mark, probably for just not being specific enough. Oh, the number of threatened species has increased more rapidly. Oh, no, I didn't. Okay, 5, yeah, because I'm a net loss of 2 marks now. Okay. Genetic bottleneck, fine. Behavioral, anatomical, physiological, anatomical. Nice, 2 marks there. Evaluate the student's conclusion. Species live in the same geographical area for Sympatric. Do I get similar niche? No, I've said different niches for allopatric, haven't I? Ooh, didn't like this one either. Didn't like this one. Evidence for allopatric speciation. Right, okay, mountain ranges, yes. The idea that the Kaka flew to the North Island. Oh, sorry, that's the wrong one. Proto-Kaka flew North Island, or that some of the Kaka returned. Oh, I mentioned that. Flew back. Yeah, that's second mark. Right, agree. Similar niches because they eat on the forest floor. Right, yeah, behavioral. Yeah, right, okay, behavioral. I get a mark. Probably it, to be honest. So I think that's a 3. Right, so I've lost 3 marks. So lack of detail here. Yes and yes. Right, we're getting there. Maltose carbon dioxide produced consistently higher. Yes. Greater volume as we go along. Difference between increases over time. Yeah, good. 2 marks there. Do I agree? Volumes produced are low. Yeah, well, they are different. Volume of carbon dioxide is the same up to 20 minutes and 90 minutes. Yes, I've given both of those. Data of the same. Okay, disagree. Sucrose increases earlier. I've said fructose is higher. Final. Okay, or reverse. Like fructose increases later. Okay, right, fine. 2 marks. Good. Named equipment with higher resolution. Okay, volumetric flask. Yes. Graduated pipette. Yes. Good. Distilled, sealed. Yes. Yes. Standard deviation. Yes. Good. 3 marks there. Oops. Go away. Nearly there. Right, body plan. So overall body. So okay, I've mentioned body plan. That's correct. Switch different genes on and off in different cells and tissues. Yes, because I talked about transcription. Cell identity. No, didn't say that. Expressed in a set order. Yes. Regulate apoptosis, mitosis. Yes. And then determine head and the tail regions where the brain will develop. Yes. Switch genes on or off in the brain. Yeah, transcription on or off. Form specialized nerve cell. Well, that's 6 anyway. Yeah. Good. Right. Explain. So the idea that archaea and eukarya are most closely related because of the DNA polymerase. Yeah, that was there. Bacteria are most closely related to archaea because of the similarities in ribosomes. Yes. Oh, I didn't mention lack of mitochondria. Yeah, useful note. Helicase provides no comparison. Yeah, okay, good, 3 marks. And then catalyzes formation of phosphodiester bonds. Yes, and breaks hydrogen bonds to allow breaking of hydrogen bonds. Good. Okay, that's it. So what's that? Net loss of 3, isn't it? So overall, how many marks was it? So I got 67 out of 70. Good, not bad. Slightly rushed it at the end, but that was fine. 67 divided by 70. So what's that? 96%. Good. Not to be sniffed at. As I said, that's it. So hopefully that proves a point that even teachers get stuff wrong. So there we go. You don't need to get every mark on every paper. 67%, as I said last year, was an A star. So there we go. Right, I'm going to stop sharing and bid you good evening.